I'm curious about how you would characterize stratified medicine, DNA sequencing.
Yes.
You've been
Yes.
at the
Yes.
forefront of these two areas.
Yes.
And when do you think, or do you have a prediction for the timing of delivery? When does this start to reach patients and alleviate the burden of disease? When does it begin to have a broad impact? I mean, the critique has been the hype and overpromise around personalized medicine.
Yes.
And it seems to be a phenomenon always around the genome. We
Yes.
overextend its
Yes.
likelihood of having impact.
Yes. So what I would say is, even today, with Mendelian disorders, we are having an impact. So if you think of the list of hundreds of Mendelian diseases that when you approach them with DNA sequencing, and I think Baylor University is one place that's done a lot, to identify the causative mutations in patient families, very limited number to date, maybe 50, that is going to grow exponentially. So I think the field of rare diseases will really explode. The challenge always is that the identification of the mutation often doesn't directly translate into what the medicine is that you need to make, and that's what frustrates many of us. But it opens up new pathways, it opens up new discovery routes, and it is important. So I think that in the next 5 to 8 years, 50% of those Mendelian disorders will be solved. You know, we will know. That may be an overestimate, but you have to think bold. Many of those, what we worry about, are de novo mutations that make the challenge more complicated and make the patient populations more difficult, where the parents don't show the phenotype and the child does. Schizophrenia is one great example there. So you're looking for those rare de novo mutations, which are a lot harder to then validate. Is this really the causative? But in common diseases, you know, I don't know if you've talked to Heidi Riom at Harvard Partners. She oversees their molecular laboratory and is really at the cutting edge, I think, of overlaying genetic sequence data into a database with phenotypic information, communicating with a physician, and really doing some fascinating pilot studies in that area. And it's really probably some of the most cutting edge that I've seen in the field. Really, really fascinating. Where a particular area of interest to her is both deafness and cardiomyopathy, or cardiac, I think HCM. And they regularly have patients that they have DNA sequence information for. As new variants emerge and new characterization of those variants emerge, they go back out to the physician, re-communicate. This variant that we saw in your genotype, we can now tell you is probably a silent, no functional consequence, or this is a new mutation that has been further characterized and is probably a causative allele. I mean, the communication back and forth with physicians is amazing, all electronically. Mm-hmm. Really interesting.
We're also very intensely curious here about the delivery of this information into the clinic, into physicians,
yes.
and into disease management.
Yes.
And the tip of the spear there, is that going to be targeted resequencing,
yeah.
exomics, or is it
yes.
going to be simply the known and highly classified mutational status?
No.
In that?
It's rapidly moving to targeted resequencing of a panel of genes, the exome, that really can then be used to translate into some decision making for patients. And I think, again, the technology has really driven us to allow us to do this more cheaply in a way that can be CLIA compliant for patients and is cost effective. So, you know, I think, you know, the ion torrent machine is really going to be a practicing machine in the clinic.
So if that's true, and this is going to be introduced to the clinic, and the clinic itself is identifying different markers
yes.
that have an implication on therapy that the individual would get.
Yes.
and they're doing this on their own.
Yes.
How does that radically change pharmaceuticals or biologic companies and how they will either develop a product or even introduce a product to market when they may not be able to influence what the clinician does anymore, that the data is actually driving the downstream decision?
Yes. Well, the way I say it is the pharmaceutical company needs to know what that information is right from the beginning. So, you know, if you have those variants and you know you want to relate them to your therapy, that sequencing needs to be part of your phase 2 clinical program. Now, the question is, and that's often a co-development strategy where you're developing what might be that diagnostic for use with the therapy. The question becomes, when you develop the therapy, you haven't done that, and the physicians out in the marketplace are doing it, and all of a sudden they know more than you know about your therapy.
So what is it about the architecture or the unknown variances in the non-coding regions of the genome that have influence on disease? I'm sure you had some
really fascinating. I mean, one of the things we spent a lot of time, because the beauty of single molecule sequencing that was far underappreciated because we just didn't have the commercial horsepower, was the beauty of the accuracy of the measurement. So in many times when you're studying gene expression differences in an asthmatic versus, you know, a non-asthmatic, when you look at RNA changes, they may be as subtle as 15 to 20%, but they point to really important pathways that you'll never see by an amplification-based method because of the challenges of doing your initial PCR. Those subtle differences are very difficult to measure. So we did a lot of characterization of the RNA. And really what we were always looking at was the entire spectrum of RNAs, not just polyadenylated RNA, which is your coding RNA. And in the field of oncology, these non-coding changes were almost diagnostic. You know, these big regions of non-coding RNAs that occurred in the metastatic population were just absolutely fascinating and could almost be, you know, useful as an indicator of the state of those cells. It was really fascinating. Unexplored world, a lot of biology. What I always worried about is you could look at those RNAs, but what do you do with those RNAs? How do you silence them? Do you want to silence them? And obviously there's been, you know, companies started around this, the world of non-coding RNA now. And I wish them a lot of luck because it really is challenging. But, you know, again, unexplored territory. The genome is unbelievably actively transcribed.
And what about the aspect of epigenomics and beyond the nucleotide control of the
yes.
genome?
What's interesting is, again, coming back to Pfizer, the real strong interest in epigenomics, epigenomic targets in oncology, the epigenomic state of tissue. I'm still trying to understand it, because I've always felt that the epigenome state of the DNA is readily interpretable in the RNA as it's transcribed and an easier measurement. But there is a lot of interest in the enzymes that are involved in modification of the genome and how that could help in oncology, but also in other diseases like inflammatory diseases. And this is an area I don't know a lot about today. But it does go to that huge dynamic state. I studied a lot of epigenomics at the level of stem cell differentiation and, you know, looking for marks on the genome that then resulted in opening up of chromatin structure and so forth. But at the therapeutic level, I'm still trying to understand it.
And maybe my last question is for where is the business opportunity in the next 2-3 years in stratified medicine?
There's many. I think it is, we still need the research that points to dissecting the heterogeneity of common diseases. So but you know, again, I think the Broad, for example, is one institution that's doing a good job with this. I think the implementation of the technologies in a clinical laboratory is an important investment area. I think it's all information. I really think it's the information that underlies all of this is just one bet that I would place.